we have David Pamies coming. He's the head of the Stem Cells and Organoids facility at the University of Lausanne, where he also leads his own research group as a toxicologist in the Department of Biomedical Sciences. His research focuses on developing advanced in vitro models such as organoids and microphysiological systems to study neurological diseases and neurotoxicity. And as a further role, he is also an expert and serves as an expert as an advisor to European and national regulatory authorities, including the EFSA, Swissmedic, and the OECD, where he contributes to the development of alternative testing strategies and science-based policy frameworks. So thank you for being here, David. So I hand it over to you. Thank you.
Well, first of all, thank you for the nice introduction. Also, thank you for the organizers to give me the opportunity to be here. And I'm very impressed that people are still here strong. I hope it will not be so boring for the last presentation. So now my goal will be to show you how human advanced models can help in the area of toxicology and drug development. And for you to understand, this is a little bit interesting field, right? Because the let's say the move or the switch to in vitro models are not coming only from the advance of the techniques, but also by the push for the regulatory agencies to do so. Just to give a little bit of a background, in 2007, for example, it was presented this report, Toxicology Testing in the 21st Century, a Vision and Strategy. This was done by the Scientific Council in the US, and the report proposed a major shift from animal-based toxicity testing towards human cell-based in vitro assays. And they argue that human biology and molecular mechanism responses and to move into these in vitro methods could help to better study human risk, do it quicker, less costly, and also use fewer animals. And of course, they already knew in that moment that will require some scientific advances and also infrastructure changes, validation strategies, and also regulatory acceptance. So they will knew that this might take some time. But if we see the efforts from Europe, we have seen that even from more than 30 years, there are some institutions like European Union for Reference Laboratory for Alternatives to Animal Testing that has been pushing into replacement, reduction, and refinement. There are other activities like the EPAA, that is the European Partnership for Alternatives Approach to Animal Testing. And we have a clear example with the cosmetic ban, where basically it was banned the use of animals for ingredients in 2004, and then, for animals to use in cosmetics in 2004, then for the ingredients that are in the cosmetics, and even after that for the ingredients that were tested elsewhere, and that was in 2013. So already you have seen that there is a push from these institutions to move into alternatives to animal testing. And even more recently, the OECD, that is the organization that developed the guidelines for industry to put compounds into the market, they also show, for example, this overview, where they are also mentioning that they would like to see more new alternative methods of NAMs in vitro and in silico methods. If we move into drug screening, this also has been a big push from FDA and you know the European Medicine Agency. In 2022, there was this Modernization Act, where it was signed by the President of the United States, where the idea that the authorities now need to be looking into alternatives to animal testing, and now they will accept non-animal alternatives for drug submission. And even more recently, in 2025, so this year, they have they have released this roadmap to reducing animal testing in preclinical safety studies, and they are trying to promote the adoption of the NAMs, but also including organ-on-a-chip, human cells, organoids, and in silico models. And this has also been supported by the European Medicine Agency. So you can see that it is not only, it is of course the advance that drive this, but it is also a special context because we have now these organization bodies pushing into moving to that direction. So I want to give you an example. I will give you 3 examples of how we can use these in vitro models for regulatory toxicity and also for drug development. And first, before I start, I want to show you the model that we use for these approaches. This model is called brain spheroids, or also we call brain microphysiological systems. And this was developed under the umbrella of Human on Chip. That was a huge project in US where it was supported by DARPA, FDA, and NIH. And at that point, in 2013, my former boss, Thomas Hartung, received some money together with Hong Yun-sung at Johns Hopkins University to try to develop a multicellular human iPS-derived 3D model that could be used for toxicology and drug development. And at that point, I was working with Elena Hopper, that was my supervisor. So we did like many others. We started from pluripotent stem cells, and then we differentiated into neuroprogenitors. And then we were using a gyratory shaking like this to aggregate the cells. And this was taken from Honegger lab, where they were using this for rat organoids. So basically we did that, and then we generate these spheroids that are in the beginning neuroprogenitor spheroids, and then we add the differentiation. And after 8 weeks of differentiation, we can obtain these spheroids. There are around 350 microns spheroids. They are quite reproducible in shape and size. And the good thing of these spheroids is they contain several cell types, and we have characterized, I think, at this point quite in deep, because we have several publications with the model. We have seen they have synapses formation. We have seen that we have glial cells. We have different neural cell types. For example, here in this picture, you can see dopaminergic neurons in red, and a general marker for neurons in green. But we also have seen glial cells, like the supportive cells of the brain. And here we have a marker that has been used for astrocytes, that is GFAP in red. And we also have a marker for oligodendrocytes. And so we were really happy that we could find all these cells together. But something that we find also quite interesting is the level of maturation you can get when you grow them in 3D. So we found out that in this model also we can produce a lot of myelin, and we can see here in green the myelin basic protein. And this was done with the neuropathology department at the University, Johns Hopkins University. We also spent a lot of work into make sure that they were quite reproducible, because in toxicology, this is a really important thing to consider when you evaluating several compounds. So we make sure that every time we produce the model, we have the same shape and then also the same size. And also we evaluate the composition of the cells by flow cytometry, image analysis, and we figured out that we have around 70% neurons, 20% astrocytes, and 10% oligodendrocytes. More recently, we are doing a study where we are in different labs that are using the model with different lines. We are trying to evaluate how reproducible it is in different laboratories and also using different lines. This is a paper that is almost ready for submission, and we have seen that in fact all the labs were able to reproduce the model, and we have with different lines you were able to produce similar neural types with small differences, for example, in size depending on the shaker that you're using. Something that is also very interesting, and I already mentioned, is the myelin information. And as you can see here, there's 3D reconstruction of the myelin around the axon, and also here electron microscopy where we can see the myelin wrapping the axons. And they also have shown to produce electrical activity. And so now I want to show you some examples of why we have been using this model and for what. One of the applications is for developmental neurotoxicity. We have spent several years working on this. Basically, there are many evidence indicating that environmental chemicals can contribute to developmental neurotoxicity and also cognitive function. So it is known that there are chemicals that might be in the market that can produce this. But there is a guidelines for developmental neurotoxicity by the OECD. So in theory, industry should do this test before putting into the market. However, the problem is that this assay is really expensive. It costs $1.4 million per compound. It takes between 1,000 and 4,000 animals and between 3 months and 2 years. So because of this, it's really impossible for industry to assume this cost and to do these experiments. And also it will be a bit crazy due to the amount of animals. So it is done sometimes when there is indication of foreseeable developmental neurotoxicity, but not routinely. And so there is a lack of information of most of the chemicals in the market, including high volume, and that has become a concern in the society. And already in 2013, there were since 2013, it has been several meetings with experts and also some agencies like EFSA, that is the European Food Safety Authority, and as I mentioned, OECD. And there have been several discussions to see how we can solve this problem. And after several meetings, it was suggested that a battery of assays, in vitro assays, that could cover the different processes in the brain could be able to solve this issue. So basically, it was they developed a battery, and this battery was developed by 2 universities and EPA, and they test many compounds, and this is something that we normally do in toxicology. We test compounds that we know, positive control, negative controls, to make sure that the assays actually can predict the toxicity of the compounds. And this was already released. So the idea is to cover different steps, like, for example, cytogenesis, neuroagro, myelin, and of course, this is a simplification of the processes. As I mentioned, this study has been published by OECD. It's called Initial Recommendation of Evaluation of Data from Developmental Neurotoxicity in Vitro Testing Battery. And here you can see a little bit how the battery looks. We have all the neurodevelopmental processes, and here we have all the assays that has been developed, and this is in humans and this is in rat. And there's also some data gap identification that was published to show where new developments need to be done to improve the battery. And one of the parts was the myelination, because there were no any assay. So because we have myelin in our model, we thought, okay, maybe we can use this for developing an assay that could be introduced in the DNT battery and make it better. So for that, we did a literature research, because there is not much about positive controls, and then we identify compounds that are known to produce myelin disruption. We generate the figures, we treat them, and then we quantify. For the quantification, and in that moment, Megan Chestnut, my student, was working on that. We took some software from Kerman and collaborators, and here I put the link in case someone wants to have a look. They were using a software in a mouse, embryonic mouse model. So basically, we just adapted to our model. You have a marker for axons and a marker for myelin. You do the binary pictures, and then it colocalizes the pixels, so it can give you an idea of how much myelin you can find. And just to give you an example, here we have a vehicle control, and here we have our marker for myelin, and then for axons, and then you can see it is quantified at 57% is myelinated. And then when we put cuprison, for example, that is known to be a myelin disruptor, we see a decrease, of course, and then we can see that obviously the color localization is reduced. So we spend a lot of time into this project, where we have 6 compounds, including a negative control. We have 2 markers, MVP and PLP1. There are 2 proteins that are known to be in the myelin. So in this experiment, we have 3 triplicates, and we took 10 pictures for each. So 1 picture per spheroid, 10 spheroids. So it was a lot of pictures, but we managed to have we managed to have a good quantification, and this was published already. However, if we want to move into toxicology, it has to be something that can be done, first of all, less biased, because here we were taking the picture, and even if you try to be completely objective, there's some risk to be a little bit biased. So we want to have something that is more neutral, and also that is for high throughput screening, right? So Now, with the support of the NRP79, we are trying to move this assay into more automatic high throughput analysis. For that, we are using two techniques. For one side, we are using flow cytometry because we have it in-house and was easy to try it. So this will remove the user bias. And then we also have a high throughput automatic confocal microscopy. So we are trying to acquire the pictures in a more automatic way. So the idea of the flow cytometry, we have it from Smirnova group at Johns Hopkins, and we used to work together. So basically, she developed a line that is fluorescence for PLP1. So we contact them, and in a collaborative way, now we're using this PLP reporting line, and we also have a line without PLP and using an antibody. And now we are testing these compounds here. I just show an example, but we have around 20 compounds with 3 concentrations per compound, and the idea is to publish this by the end of the year if everything goes well. Regarding to the high throughput confocal microscopy, we are using a little bit different approach, and this is because the free floating is a nightmare for image analysis. So we decided to move into these 3D grids from Sun Bioscience and now from InSphere, where we grow the spheroids into these hydrogels micro wells. And the good thing is, after 8 weeks, you can see here all the micro wells. This is allowed the microscope to find similar focal plane, and even if we have autofocus, it improves a lot when we have this into the grid that when we have just lying into the bottom. And with that, we are doing the same thing. Here is the total fluorescent, but now we are using AI software, AI-based software in Encarta package to try to quantify better. And we are also planning for next year to introduce iPS-derived microglia that is already developed the protocol in our laboratory. Another application I want to show is regarding to glioblastoma. Glioblastoma is the most aggressive and common primary brain tumor in adults, and they have very low survival. They have is very heterogeneous and have a multigenetic mutation that make them quite resistant to therapies. So we were trying to figure out if we can develop a model that could be used for drug screening and maybe personalized medicine in the future. So basically, here we have a project. Here we have a project with together with Alfredo Quiñones that is from Mayo Clinic, where we introduce a tumor into our brain spheroids. You can see how the tumor grows over time. The tumor was marked to be fluorescent, so it was facilitating the analysis. We collaborate with Micromatrix. It's a company that basically what we did is to put one spheroid with the tumor in each well with the different treatments. And after they embedded into this spheromatrix marker TMA, that is like a wax, and then they do a lot of sections, and then you can do several stainings for the treatment. And they have also an in-house software that can help you to analyze and identify the areas that you want to study. In this case, for example, the tumor or, Ki-67, that is a marker for proliferation and things like that. Just to give an example, here we have a treatment with doxorubicin. So here you can see clear the tumor, and when we are adding the compound in the concentrations, you can see how slowly, when we increase the concentration, we see a decrease of the tumor. This is really good because you can see not only the effect in the tumor, but also in the healthy line. So it can help you to define a therapeutic concentration. And this has been already published. And now we are moving forward with another molecule with Sukri Habib and Pierre Becquart that now we are trying to develop in our facility. The last thing I want to mention, and I have only 2 minutes, so I will not go in deep. Basically, we also work together with EPA. They have an assay where they use rat cortical neurons assay for neurotoxicity, and they use it for screening compounds in US. So basically, together with Tim Schefter, we figured it out. We wanted to know if we can substitute the rat cortical neurons for the brain spheroids that we have. And so for that, we use a multi-electrode array. and basically has 26,400 electrodes. So we can see the organoids, and then we can select the areas, and then we can extract information from each brain spheroid. And we use same compounds that they are using in EPA. So they have a list of compounds they have already been tested, and then we test the same ones, and then we compare, and we find out that actually the data was quite similar. So we hope that in the future this can be substituted, and this has also been published. With that, I want to thank everyone. Of course, many people was involved in these projects and many funding bodies. I hope I mentioned at least the key people in the projects. And just, I could not avoid, I know it's not the reason for the presentation, but I just want to mention that we have this facility at UNIL where we help scientists to generate lines, characterize the cells. We have a really cool machines like spatial transcriptomics. We do trainings, and also we do organoids on demand. If someone is interested in a specific organoid, we create them for them so they can use for their research. And with that, I think I did almost 20 minutes. So I'm done. Thank you very much.
Thank you very much, David. Yeah, so you were good in time. Thanks. I'm pretty sure there are some questions I saw in the chat, right?
Yes, there were some questions. Also question like for sometimes you have tumors which spread into the brain, like leukemia, and the question was if you could also check for the neurotoxicity of a compound used against this leukemia.
Yes, exactly. So the idea to have both is to not only, because there are many people using tumoroids now where you have basically the tumor tissue. The idea is to combine with healthy tissue, so then you can see also if these compounds might affect also the healthy cells per se.
Mm-hmm. Okay. And for the remyelinization assay, there was a question, is it a permanent effect if it stops by cuprizone, for example, or if you remove a cuprizone, does it go on?
So very good question. I think for at the moment we have not studied remyelination, so we are only interested to see if compounds are normally environmental compounds, like pesticides, might affect the myelin formation during the development, right? But I think if someone is interested, it could be nice to use also the model to study remyelination after the exposure to some compounds, maybe with some drugs that could help to this remyelination. But we have not done it at the moment.
And maybe at the end, a technical question, but it's coming up all the time when it's about organoids, and I think it's been 3 times in this session already. Like people ask, what do you do because there is no endothelial cells in the organization? There's no vascularization.
Yeah, so this is really common in the facility, from the facility perspective. This is a really common problem, right? When you have big organoids, then you start getting necrotic centers, and it difficults a lot, especially in toxicology, right? Where you don't want to see, cells dying for other reasons. You want to see with the compound, right? So for that project, that's why we are using spheroids instead of cerebral organoids, for example, because it has been shown in several publications that if you keep the tissue lower than 500 microns, you normally cannot observe these necrotic centers. So in our case, we have around 350, 400 microns, and we have not observed this problem into the model. But it is true that if you go to higher models, bigger models, you most likely will have these problems of, necrotic centers, and this is a big issue for toxicology, of course.
Maybe it could be even a good thing, because you have sort of zones. Now you could see an oxygen-rich zone and an oxygen-lower zone. So it might not be a bug, but a feature, maybe, of your organoids.
There are some people that have been trying to develop the vasculature into the organoids. So there are some people working into that direction. At the moment, I don't think there is any super amazing model where you can see fluid going through the vessels, things like that, but it is going into that direction.
Okay. Thank you.
Okay, is there another question, Joachim, or not?
Yeah, it was similar to like with oxygen again. If you check the transcriptomics for the center versus the outer region of your organ.
Say again.
If you did transcriptomics of the different regions of the brain spheres.
So when you use, for example, cerebral organoids where you have, different regions have different structure and different populations, right? So it makes sense to do this type of things. In our case, the spheroids are really homogeneous, right? You have different cell types, but we don't have region distinction. So for us, in that sense, it doesn't make much sense. We didn't see any differences whatsoever with this single cell sequencing, and everything seems to be quite homogeneous. We have not done a spatial transcriptomic on those yet, but we also don't feel that it's a need because it's quite homogeneous when we observe the picture we had done cryo sections, and everything seems to be quite similar. And in other sense, when you have something complex with different structure, then it makes more sense to study these type of things.
Thank you.
Okay, then I think we close your presentation here. Thank you very much, David,
You're
for
welcome.
showing your research.